Hey everyone, and welcome to this explainer. Today, we're essentially putting on our detective hats to crack the ultimate biological code, human inheritance. Using the concepts laid out in SPM Chapter 11, we're going to figure out exactly how physical traits get passed down from generation to generation, and how one tiny microscopic typo can literally change everything. Let's dive right in. So here is our roadmap for demystifying all of this today. First, genes, alleles, and traits. Second, solving Mendelian crosses. Number 3, human inheritance patterns. 4, decoding family pedigrees. And finally, chromosomes and sex determination.
Okay, let's jump into Section 1, genes, alleles, and traits.
To do our genetic detective work, we first need to figure out where our clues actually live. And that brings us to the locus. You can think of a locus as the exact physical street address of a gene on a chromosome. When we're tracking inheritance, we're basically just watching who moves in and out of these very specific microscopic addresses.
Check out these homologous chromosomes side by side. As you'll see, they carry alleles for the exact same gene at identical loci. See that capital H? That's our dominant allele. And sitting right across from it at the exact same address on the matching chromosome is a lowercase h. That one is our recessive allele.
Knowing this little detail lets us classify these allelic combinations with total precision. So, if the alleles at the loci are identical, say capital T, or maybe small b, we call that genotype homozygous. That also makes them purebreds. But if the alleles are different, like capital D, small d, they're heterozygous, or what we call hybrids. Pinpointing these specific pairs is absolutely crucial. It's our very first step in predicting how traits get passed on, which is actually a giant puzzle that was first solved by a pretty famous monk.
Which brings us to Section 2, solving Mendelian crosses.
Gregor Mendel left us with 2 golden rules. His first law, the law of segregation, essentially means a trait is controlled by a pair of alleles, but a gamete only ever inherits one of them. Then there's his second law, the law of independent assortment. This means that during gamete formation, alleles from different pairs just mix and match completely at random. And that randomness, it's completely key when we start tracking multiple traits all at once.
Let's look at this monohybrid cross. Monohybrid just means we're tracking one single characteristic, in this case, seed color. We're crossing a purebred yellow pea with a purebred green pea. Look at the result. The entire first filial, or F1 generation, produces exclusively yellow seeds.
So, here is the big question. If both parents were purebreds, and they contributed equally to the offspring's genetic makeup, why in the world do the green traits completely disappear in that first generation? Where exactly did the green go?
Well, the answer lies in the dominance principle. The yellow trait is the dominant allele here. Whenever a dominant allele is paired up with a recessive one, like our green allele, it completely masks or suppresses the expression of the recessive one. The green genetic information is actually still there, hiding out in that heterozygous genotype, but only the dominant yellow phenotype is visibly expressed. Pretty wild, right?
Now things get really interesting when we upgrade to a dihybrid cross. This is where we track 2 independent traits at the exact same time, like fur color and tail length in rodents. Just look at the resulting offspring data here. We've got a mix, 13 with black fur and long tails, 12 with black fur and short tails, 10 with white fur and long tails, and 10 with white fur and short tails.
When we crunch those numbers from the module, a massive clue pops up. It's a mathematically distinct 1-to-1-to-1-to-1 phenotypic ratio. For us genetic detectives, this very specific ratio points to one exact scenario: a test cross between a parent who is heterozygous for both traits and a parent who is homozygous recessive for both traits. It is Mendel's second law, perfectly visualized.
Moving right along to Section 3, human inheritance patterns.
A classic real-world example of Mendelian inheritance in us humans is tongue rolling. Seriously, try it right now. Can you roll the sides of your tongue into a little tube? If you can, you've got the dominant trait. If you can't, it strictly means you possess the homozygous recessive genotype for this specific characteristic.
So let's map this out. If a tongue-rolling father who carries the recessive gene, meaning he is heterozygous RR, has a child with a non-rolling mother who has to be homozygous recessive, or R, what happens? Well, the offspring will follow a strict, completely predictable 1-to-1 ratio. Every single child has exactly a 50% chance of inheriting the ability to roll their tongue and a 50% chance of not being able to.
Okay, on to Section 4,
Decoding family pedigrees.
Take a look at this pedigree chart. It maps out hemophilia, which is a condition where a person's blood fails to clot normally. Genetically speaking, it's super fascinating because it's a sex-linked recessive disorder. The gene that controls this blood clotting factor is located entirely on the X chromosome. The Y chromosome is physically shorter and literally just does not carry an allele for this trait at all.
So, looking at the affected male in the third generation, individual R over there, we have to ask ourselves, based on the rigid rules of sex-linked inheritance, what must his specific genetic makeup be for him to express this disease?
Well, because males have an X and a Y chromosome, they only have one single X chromosome to rely on. Because of that, receiving just one single recessive allele from the mother results in the XHY genotype. There's no dominant allele on a second X chromosome to swoop in and mask it, so the disease is fully expressed. And honestly, this is exactly why sex-linked recessive conditions show up way more often in males than in females.
Finally, Section 5, Chromosomes and Sex Determination.
This karyotype reveals a really clear case of a chromosomal abnormality. Notice the visual arrangement of the chromosomes into homologous pairs. But if you look closely at the sex chromosomes down there, we see a clear case of non-disjunction. That basically just means the chromosomes failed to separate properly when the cell was dividing, which in this case results in a genetic condition known as Klinefelter syndrome.
The defining diagnostic number to remember here is 47. A normal human somatic cell has 46 chromosomes, but this individual has an abnormal chromosome count. 44 normal autosomes plus an X, another X, and the Y sex chromosome configuration. Because that Y chromosome is present, the individual is male, but that extra X chromosome leads to sterility and the development of female secondary sex characteristics.
But under normal somatic conditions, sex determination is basically a straightforward biological coin flip. The mother provides an ovum that always carries an X chromosome. The father's sperm, produced through meiosis, carries either an X or a Y chromosome. The fusion of those haploid gametes guarantees exactly a 50% probability of having a boy, with 44 autosomes plus XY, or a girl with 44 autosomes plus XX.
We've covered a lot of ground today, tracking alleles from their physical locus all the way up to complex human chromosomal patterns. We'll wrap up our explainer today with this thought to chew on. As genetic mapping technology continues to advance at lightning speed, how might decoding your own family's genetic history, maybe mapping out your own hidden recessive traits, predict your future health? Keep observing, keep questioning, and keep exploring the science around you. Thanks for joining me.